years, but we were fortunate to get our keynote today, Dr. Mario Capecchi, and I've been a big fan of his for many years and never met him until this morning when I discovered he was here dusting on tables before anybody else got here this morning. And as Dr. Capecchi probably doesn't need much of an introduction to this group, he is a Nobel laureate from 2007 who shared the Nobel Prize for creating the transgenic mouse. And he told me this morning that the National Institutes of Health has reminded him repeatedly how much money they've costed them. Because the transgenic mouse population, as you realize, is up in, I think, the 12,000s now. Dr. Capecchi, if you go to his Wikipedia page, has a very interesting childhood, which we won't go into in this introduction. But he developed at Antioch College in Ohio, which he tells me was an interesting program because you spend half your time in school and the other half your time in laboratory, or in his case in laboratories, or the other half your time working. In his case, his work time at Antioch was actually at MIT. So the time he finished his undergraduate degree and he was a physics major, he'd already had some exposure to MIT, which I think at that time was probably a different place. So he came to Harvard to study biophysics in the biophysics department. And yesterday, he returned to that department to give a talk. He said it was the first time he'd been back there since he was in that department. He graduated from there in '67, was immediately scooped up by Harvard Medical School in the biochemistry department, and in 4 short years became associate professor. And in those 4 short years, the work that he focused on was protein synthesis. Much of what we know about protein synthesis beginning and ending was work that Dr. Capecchi did in those 4 short years. And all of a sudden, he went to Utah. And I've always wondered, my gosh, what would cause you to go to Utah? Is it because you're an avid skier? Is it because you really converted to Mormonism overnight? What exactly would recruit you to Utah? And he may expound on this a little bit more, but he felt that it was at that time a much more collegial place where you could really build an infrastructure of people who wanted to work together rather than people who were whispering so that the person in the lab next door didn't know what you were doing. So that sort of speaks at what was going on at Harvard Medical School in the early 70s, which I know to be true. And probably hasn't changed a whole lot. And so while we get Dr. Capecchi's slide organized for you today, or his talk, he's going to talk to us on past, present, and future on his perspective, and he is still very busily running his laboratory. So this is work that is not retrospective, but is ongoing and prospective.
Well, it's a pleasure to be here and share our work with you. I'm going to take you a little bit in a different direction from stem cell biology. We do work in stem cells. But what I was going to do today is just give you a couple stories from the lab, a progress report on those particular projects we're working on. We work actually on many different projects, so this sort of gives you a flavor. I'm going to describe 2 stories. First of all, our patients are mice. That's what we work with. And what we do is model human diseases in mice. And I'm going to talk about, first of all, modeling sarcomas. And then second, I'm going to talk about modeling neuropsychiatric disorders. And the first thing you might ask yourself is, why model sarcomas? They're not as common as carcinomas. However, they affect a critical population, that is, children and young adults. Secondly, they're extremely aggressive. For example, the particular sarcomas we've modeled now, we've modeled 4 different ones. The prognosis is that after first presentation, 80% of those children are going to die within 5 years. And that hasn't changed in the last 3 decades. So that tells us that current methods of surgery, chemotherapy, and radiation are not effective. And we have to find a new way. And the only way I know is to actually start understanding the cancer in molecular detail and then develop a therapy based on that understanding. They're not as extensively studied, and that's simply because they're not as common. Pediatric oncologists see them, but most cancer biologists don't see them. However, they have a number of really advantages in terms of study. And the first is that we think we know from many of them. I should also point out that in biology, you can never make generalizations that will cover everything, okay? There will always be exceptions, and so remember that as I talk. However, for most sarcomas, it's not true for all sarcomas, but for most sarcomas, the initiating event is the breakage of two chromosomes and then the rejoining of opposite partners. So now the information base is still the same, except at that junction. And at that junction, you'll have a new gene being made, which is half coming from one chromosome, the other half coming from the other chromosome. And obviously, there's also a reciprocal since it's a reciprocal recombination event, you'll have the opposite, where you'll have the beginning of the other chromosome, the gene on the other chromosome, being hooked onto now at the other end of the other chromosome, okay? So you have essentially four products, you've two new gene products, and if you lost that, the alleles are involved in making those gene products. Okay? So we think we know the initiating event. And then, and if you're a modeler, you like to start at the beginning. So that's one of the reasons for working in these cancers. And then finally, they're not as complex as carcinomas. And that is because genome instability is not the hallmark of the majority of sarcomas. Okay? After you have genome instability, you have thousands upon thousands of events have occurred in the cell, and it's very difficult to ascertain which ones are contributing to the cancer, progression of the cancer, and which are not. Okay? Whereas in sarcomas, often the only aberrant chromosomal event that one sees is the translocation itself, and that's it. Okay? So it makes identifying all the subsequent steps required essentially for progression of the cancer much easier. Okay, so those are the reason we're looking at it. And I finally should point out that an additional property is that the translocation product itself is not only involved in initiating the cancer, but often also in maintenance of the cancer. So it's a major driver in the cancer. And once you have a major driver, then you obviously have a target for therapy. Okay? So those are the reasons why we're working on sarcomas. And the one I'm going to be talking about is synovial sarcoma. The other thing I do is always say we. And we is always the royal we. It's actually graduate students and postdocs in the lab, and I'll give credit where credit is due. Now, this is what we knew when we started this particular model, and that is that synovial sarcomas arise near joints. And in fact, that's where the name is derived, because synovial tissue is also associated with joints, and therefore the name. And that turns out to be a misnomer. They're histologically complex. There are three histological subtypes. There's what's called monophasic, which are spindle cells, mesenchymal cells, biphasics, which include also spindle cells as well as ectodermal cells, and then finally poorly differentiated, which is common to many sarcoma, which are small cells with large nuclei and scant cytoplasm. So those are the histological subtypes in humans. And this is the axial fusion gene. It's a breakage of chromosome 18 at the SYT gene, also known as SSA18, and a break on the X chromosome known as SSX. Okay. The SSX genes are actually 9 in humans and 12 in mice. However, in human cancers, you only see SSX1 as a partner or SSX2 as a partner, and sometimes, rarely, you see SSX4, and none of the others are ever seen in synovial sarcoma. Both of these genes are chromatin remodelers. SYT, also known as I just said, SS18, is associated with activation complexes, and SSX is always associated with repressive complexes. Okay. Neither have a DNA binding domain, so they have to then associate with a transcription factor that binds to the DNA and recognizes a particular gene. Okay, this is the fusion gene. It contains almost the entire SYT gene and half of the SSX gene. It contains the activation domain of SYT and also the repressive domain of SSX. So this is a schizophrenic molecule now, capable of both recognizing repressive as well as activation domains with respect to chromatin remodeling. And here's the model, okay? And depending on the expression pattern of the genes involved, we use different models. In this particular case, space was controlled by SYT or SSAT, okay? And it's ubiquitously expressed in all cells of the body, so we didn't have to worry about space. Okay? And so what we could do is go to another gene that is also ubiquitously expressed, and that's the ROSA locus. Okay? And into the ROSA locus, we put in a cassette, which has a cDNA from the fusion gene. Okay? And the first fusion gene that in this case, it's Malay Haldar, a graduate student in the lab, isolated from the human tumors, happened to be SSY, SYT, SSX2. So that is that cDNA. Then we have an iris, which is a ribosomal reentry site that allows us then to make multiple proteins from the same transcript. And into that, we put EGFP, so those cells are capable of turning green, okay? Fluorescently green. However, between the promoter of the ROSA locus and this cassette, we put a stop sign, flank those with LOXP sites, and then LOXP sites are sequences which in the presence of CRE, which is a recombinase specific for those LOXP sites, will mediate a recombination between them. And if those two LOXP sites are in the same direction, when CRE activates recombination, it excises a stop sign and then turns on expression of the cassette, and then you make the fusion gene as well as turning on the cells green, okay? So by controlling where CRE is expressed, we can control where the fusion gene is made. Okay? So that's the model. So the first thing we asked was simply what happens if you make the fusion gene in every cell of the body? So for that we used HPRT CRE. So we have one mouse has the CRE driver, in this case HPRT CRE. The other mouse has the cassette that I just talked about. And so now we express the gene in every cell in the embryo. And what happens is you get a green blob. And that's shown down here. It's green. That's good. That means the experiment worked. The fusion gene is being made. However, it has the three tissues, the principal three tissues of the embryo, but the tissues are completely disorganized. And so then the embryo gets quickly absorbed. Okay? So this tells us that the fusion gene is very cytotoxic with respect to development, okay? And no tumors. So but that was important information. So where do we go next? We decided to look at muscle. And the reason for looking at muscle is two reasons. One, I already mentioned that the tumors have mesenchymal cells in them, suggesting muscle. Secondly, every joint essentially is associated with muscle. Okay, you want to be able to move that joint. Okay? And so we looked essentially at every cell lineage that goes to make muscle. Okay, and first of all, we look at muscle progenitor cells, PAX3 CRE, PAX7 CRE, cross it to our mice. Again, embryonic lethality, no tumors. Okay. So then we decided, well, we'll go all the way to the opposite end of the spectrum and look at fully differentiated myotubes. Okay, and for that we used MIF6 CRE, a marker of differentiated myotubes. And that is shown here. We'll do that, and here we got a surprise. We got a myopathy, and a classic myopathy. That's shown here. So here are the two controls, longitudinal sections and cross sections of muscle fibers. And here is in the presence of the fusion gene. You can see smaller fibers, wavy fibers, and all the nuclei lined up. So now I'm going to take you back to embryology. Remember that you make muscle in two waves. Okay? You first pattern muscle from the somites, okay? and then all the muscles are patterned, and then you start making the bulk of the muscle. In humans, this starts about 2 weeks after birth. In mice, it starts about 1 day before birth. And this involves satellite system. Okay? So satellites are then making the bulk of the muscle. It's all been patterned. Satellite system is also important. For example, if you exercise too much, you destroy some muscle, then satellite system is used to regenerate muscle. Okay? And a hallmark of regeneration is that all the nuclei are lined up. Okay? So that tells us that there are cells dying and then being replaced by satellite cells. And this continues for quite a while until the mouse gets a myopathy, when it just can't keep up with it anymore, and the mouse finally dies, okay, from the myopathy. Now, why is this interesting? Translocation, the frequency of translocation is enormously high, both in the soma as well as in germ. -3. That's the frequency of generating a chromosomal reciprocal translocation. In your lifetime, you have about 10 to the 17th cells, 10 to the 14th translocations. Some of these translocations do bad things, and some of them to cancer, a lot of sarcomas, each one a different translocation. Okay? However, with cancer, it's easy to see, because you have the cell essentially is a clonal population and expands. And so then it's easy to see that there's a translocation in that cell population. Until deep sequencing, we could not detect other kinds of translocation in other pathologies. And now people are starting to do that. What are they seeing in the myopathies is translocations. And what I would argue is you're going to start seeing somatic mutations in many other diseases as a consequence of translocations, now that you have the tools to see it. Okay? So just keep that in mind. But let's move forward. Okay, so far no tumors. So where could we go next? And there we go to myoblasts. And here we had a choice. There are 2 genes that are involved in specifying myoblasts, MyoD and MIF5. And we decided to make MIF5, that is, Malay Haldane made MIF5 Cree, crossed it to our mouse, and we get tumors. And they're in the right place. They're always associated with joints. Okay? And the penetrance is 100%, and in fact, the mice get multiple tumors as a consequence of activation of this gene in myoblasts. They proceed all the way through metastasis. The pattern of metastasis is very similar to what we see in humans. And here we're showing a metastasis, for example, a lymph node in the cerebellum. Why do we think those are metastases? They're much smaller than the parental tumor, and they're in tissue that never express MIF5. With respect to histology, we gave the tumor material, the mouse tumor material, to a histologist that works in synovial sarcoma in humans, double-blind. She couldn't distinguish the mouse material from the human material. We also get 2 histological subtypes. We get monophasic and biphasic. In humans, if SSX1 is the partner, at first presentation it's primarily biphasic. If it's SSX2, it's primarily monophasic. We say a 10-to-1 bias for monophasic because we were using SSX2 as the partner. Markers. There are over 25 markers that have been identified specific to synovial sarcoma. We looked at all 25. They're all expressed in the mouse tumor. For example, you can see ones that are keratins are in the ectodermal cells. Myogenin is interesting. Okay, that's a differentiated muscle tumor, muscle gene. None of those genes are expressed in this tumor. Okay, and that's why it's never been recognized as a muscle tumor in humans. BCL2 positive, 97% of human tumors express BCL2. These tumors express high levels of BCL2. Vimentin, a mesenchymal marker expressed in tumors, highly expressed in the mouse tumor. And we've done gene expression-wide analysis, so with thousands of genes, and the fingerprint, and now people have sequenced many synovial human sarcomas. It has a very defined fingerprint, and these group essentially only with synovial sarcoma. So with respect to the expression pattern of thousands of genes, now it still looks like human synovial sarcoma. Now here was a surprise. I mentioned that GFP is part of that cassette. Okay, so we expect the tumor to be green. Okay, but what's out here, this is the muscle, and that autofluoresces. This is essentially autofluorescence, and this is a tumor arising out of the muscle tissue. Okay, we expected this to be green, because remember, when you make a myotube, you take thousands and thousands of myoblasts and fuse them together to make a myotube. Okay, so we expected actually all the muscle to be green, and it's not green. Why is that? To identify that, we simply looked when muscle is just starting to begin to be formed. That's around E11.5, E12. Okay, so here you can see muscle starting to be formed. However, the green cells are there, but they're not participating in making the myofibers. Why are they not participating in making the myofibers? Massive cell death. Everywhere the fusion gene is being expressed, the cells are dying. This is tunnel assays. You can see lots of red cells everywhere, except wherever there's cartilage. So here's a rib, and what you can see that right along the rib, the cells are still there, they're dividing, and they're happy. Okay. So it looks like the rib is producing a factor which is allowing survival of those cells, then to divide, now accumulate secondary events, genetic events that allow then progression of the cancer. We're very interested in trying to identify that particular factor. Now, the other thing that was strange, and remember, again, I mentioned that when you make a muscle fiber, you fuse thousands of myoblasts all together, and yet these mice are running around, they had muscle. But I also mentioned that there's redundancy between MyoD and MIF5. Okay? But there's two types of redundancies. You can have the same gene expressed in the same cell doing the same thing, or you may have separate lineages. Okay, so then if we would kill one off, then the other would still be existing. Okay, and we can test that simply by putting diphtheria toxin again in the rosa loca, put a stop sign, and now cross it to our MIF5 cells, a MIF5 Cre, okay, and then kill every MIF5 expressing cell and ask what are the consequences. And here are the consequences. Let's look at just this right here. Here's in the absence of Cre. Here's in the presence of Cre. We're obviously killing all of those myoblasts, but there's still muscle here, okay? If there wasn't muscle, what you would see is a big concave space because this is all muscle. Okay? So the muscle's still there, and then if we look at the time when we're starting to make muscle, what we can see is the MIF5 are starting to come up. Those cells are dying, and eventually if we went out here, they'd be completely dead, whereas the MyoD cells, expressing cells, are now taking off. So they compensate and thereby allow essentially the mouse to have muscle, run around, but it still develops then tumors. Okay? So that tells us that the lineages are actually independent and can compensate for each other. Okay. So in summary of this part, what I've shown you is that we have a cell of origin. I should say it's not the cell of origin. We simply showed that it is a cell of origin and likely to be a cell of origin also in humans. Okay. So now let's look at mechanism. What is the fusion gene itself doing? Okay. Now remember that these proteins don't have a DNA binding domain. So the way to approach this is to make an epitope exactly at that junction between those two different gene products that will be unique to that protein. Okay? So we make an antibody, then use that to precipitate our proteins, and then what's present in that complex. And this was done in collaboration with Torsten Nielsen of British Columbia. And you get a lot of bands. Okay? And so this is simply a gel of those proteins. We can isolate them, put them on mass spec, and identify what the proteins are. And I'll look at just 3 of those bands. One is the fusion gene. The other one is ATF2, activation transcription factor 2, and the other one's called TLE, transducin-like enhancer of split, which is a Drosophila, initially Drosophila gene that's associated with essentially inhibition complexes. What TLE does is to recruit polycomb group 1 complex and then histone deacetylases and then turn off target genes with respect to those proteins. Okay, ATF2 is an activator protein. It binds to cyclic AMP responsive element, maintains a status essentially with respect to metabolism, and then in the presence of cyclic AMP, when things are terrific, turns on those set of genes. Okay? Many ATF2 target genes have been identified, and I'll go into that. So this simply shows that in a cell line that doesn't have the fusion gene, if we add it, and it does express ATF2 and TLE1, we make a complex. This is simply a glycerol gradient, big complexes sediment at the bottom, and small things sediment at the top. And in the absence of the fusion gene, those protein products don't form. So that says that essentially the fusion gene brings those proteins together to form a complex, okay? Which was consistent also with immunoprecipitation. Here we're doing is simply making deletions across this fusion gene and asking where does ATF2 bind, where does TLE bind. ATF2 binds at the amino terminus of the fusion gene. TLE binds to the carboxy terminus of that fusion gene. Here we show that we can disrupt the complex just by using interfering RNAs. And in this case, interfering RNAs against the fusion gene itself. Again, a glycerol gradient. In the absence of, just as a control interfering RNA, you don't disrupt the complex. As soon as you add the interfering RNAs, then you break up that complex. And from that, we've made a model, essentially, where you have ATF2 binding then to cyclic AMP responsive elements. To it binds the fusion gene, which then recruits TLE1, which then in turn recruits polycomb group complex 1 and histone deacetylases, and turns off all ATF2 target genes. And in this case, for example, EGR1 is a tumor suppressor that's being turned off and contributing to tumorigenesis. And here we show that if we disrupt the complex using any siRNA against any of those possibilities, the fusion gene, ATF2 or TLE1, we reduce growth of human cell lines, cancer cell lines, synovial sarcoma cell lines, as well as mouse cell lines that we generated in the lab. Okay, so growth is dependent on that complex. So is plating efficiency, and so is induction of apoptosis. So that is that this fusion gene is also required for maintenance of those cell lines in culture. Okay, and here's simply a heat map of expression analysis looking at 10 different target genes, defined target genes of ATF2 compared to other sarcomas. And what you can see is that indeed all of those target genes are being turned down in synovial sarcoma. And the red are the proteins that are involved in making the fusion complex. Okay. Here we're looking at immunoprecipitation, then sequencing to see where is the complex on the DNA, and it's right at the, in this case, it's EGR1, the tumor suppressor gene, and it's sitting right on the cyclic AMP responsive element. We've done that for all the other target genes, again, redefining that those are the target genes, and they're being specifically turned off in these cell lines. And here we're showing you essentially that if we interfere with that complex, now those target gene expression goes back up again. Okay? Now, that's suggested because one of the inhibitor components is histone deacetylases, that we could use inhibitors through deacetylases to manage this tumor. Okay, and here we're using that. Romidepsin is a specific inhibitor of histone deacetylases, and what we can see in the presence of that, we disrupt the complex. Okay? And here, the disruption of the complex is at that interface between the fusion gene and TLE1. And this suggested that we could now possibly do, well, here's one more experiment, where we can use the two different deacetylase inhibitors to show that now, in the presence of those inhibitors, now we turn on the target genes of TLE2, okay? So that essentially shows that we can disrupt that expression pattern, and suggested essentially that these inhibitors may be effective with respect to management of human synovial sarcomas, and now there are 2 clinical trials going to test that hypothesis. Okay. The other thing I've mentioned is that BCL2 is also highly expressed in these tumors, and people are making BCL2 inhibitors. Okay, for example, for small cell lung cancer, and what they find is that it works initially, and then there's reversion. Okay, so then the inhibitor no longer works, and the reason the inhibitor doesn't work anymore in these cases is, for example, one class of inhibitors are made against what's called the BH3 domain, which is present on BCL2. However, there are other members of the BCL2 family that do not have the BH3 domain, and it's those that essentially now are expressed in the tumor and thereby make those inhibitors no longer effective. Okay? And in particular, MCL1 is a BCL2 inhibitor, a family member, and BCL2A1 are also members of that family that don't have BH3 domains and therefore provide an escape route for the tumor. However, we noticed that in synovial sarcomas, those particular family members are specifically turned off by the same mechanism that I've just described. Okay? And here we show, for example, with MCL1, that expression in synovial sarcoma is much lower compared to, for example, the original muscle tissue with respect to BCL2 expression of those family members. Okay? So that suggested that synovial sarcoma should be especially sensitive to BCL2 inhibitors and not be able to revert and become resistant to this inhibitor. And indeed, they are extremely sensitive. Here we're showing essentially a human cell line, mouse cell line, very sensitive to low doses, just 2 weeks exposure to the BCL2 inhibitor. And more interestingly, doxorubicin is the one molecule that's actually used clinically now for synovial sarcoma. And what we can see is that sensitivity in combination with doxorubicin and the BCL2 inhibitor is enormous. Okay, so a very rapid response. So again, now those are going into clinical trial with children with synovial sarcoma. And we can also show it in vivo in our mouse model. We can put in the inhibitor, and what we can see is reduction in the number of tumors that we see per mouse, as well as tumor mass after just a couple of weeks of exposure to the BCL2 inhibitor. So that finishes up that aspect of the story. And now let me turn over and look at neuropsychiatric disorders. So one thing you might ask is, a mouse is not likely to reflect human behavior. And what I'll argue is that they actually look and behave quite similar to us. Okay. We got into this by serendipity. Our laboratory has been interested in Hox genes. And I think all of you know that Hox genes are important for establishing body plan. Make sure my head is up here, my arms are at this position, all the organs inside my body are here, for example, my heart, liver, and so on and so forth. So these genes essentially give positional value. They're all transcription factors turning on hundreds of different genes, but they give essentially positional value along the different axes, whether it's the main axis or distal axes, okay? Formation of limbs, for example. But we've also compared it now to Drosophila. What you can see, there's a quadruplication of the number of Hox genes and extension of the number of ABDB genes in humans. And in mice and humans have the exact same pattern of Hox genes. Okay, so we've inactivated all of these and looking to see what happens. And the gene that I'll describe right now is HOXB8. And in this case, is Joy Greer, again, a graduate student in the lab. She inactivated HOXB8. She looked at it and it seemed pretty normal. Okay, so the other thing she said, well, okay, let's look at its behavior. Since mice do most of their behavior at nighttime, she used infrared cameras, took movies over a 24-hour period, then asked, what is the mouse doing? And she looked at everything that the mouse ever thought about doing, and what they do different is groom. They spend a lot more time grooming. Okay? And all animals groom. C. elegans grooms, Drosophila grooms, mice groom, we groom. Okay? And the syntax in all mammals is quite similar. You go into the shower, you lather your hands, start working on your head and work down your body. Okay? And that's what's shown here. The mouse lathers its paws and then works on its head and works around and ends up on the tail. Okay? In these mutants, the syntax is not changed. It's identical. What's different is they spend more time grooming, and they also, each bout of grooming is longer. Okay? So this is shown, for example, here we have pairs, SIB pairs, okay, and they're also matched for sex. And so the black bar is a HOXB8 mutant, a gray bar is a wild type sibling, okay? And what you can see is if you average over lots of different mice, the mice then spend the same amount of time eating per 24-hour period, okay? And she looked at running around the cage, walking, sleeping, drinking, eating, building nests, walking upside down in the cage, so on and so forth. Everything was identical except for grooming. And the mutant mice spent twice as much time grooming as normal mice. Okay? Quite reproducible, very robust. Okay. We can also induce grooming. You can take a sprayer, a water sprayer, spray the mouse with water, and then they start grooming like crazy. Okay, and then, but this is done, actually we cross this mouse to a real wild type mouse, that is one out in the field. What we wanted to see is its genetic background critical for this behavior, and what we can see, again, those mice, as long as they're mutant for HOXB8, spend twice as much time grooming, independent of genetic background. Now I told you one slight error. Okay? If over a 24-hour period you do too much of one thing, you've got to take it away from something else. And what these mice take it away from is sleeping. So they spend the same amount less time sleeping. Okay? Now the behavior is pathological. I'll show you a movie to illustrate this, and that's shown here. So this mouse keeps grooming and grooming. And what you'll see is when the mouse turns, it grooms one area so much that it actually removes that body hair. There, so it's removing all the body hair. Then it continues to groom and groom until it has lacerations and it has to be put to sleep. Okay, this is similar to a condition called OCD, spectrum disorder, trichotillomania. These are humans that essentially pull out all of their body hair, no matter where it is. Okay? And the children, essentially what they have to do is put gloves on them to prevent removal of body hair. It's compulsive. Now this is an interesting experiment. We put a wild type mouse in the same cage as a mutant mouse, and it grooms it and pulls out its hair, but the pattern is opposite, okay? Before I showed you it was removing the body hair, what we call the ventral side, that's what's exposed to it, whereas now it's pulling it off the back. Now why is this interesting? This is interesting because we don't think another mouse, a mutant mouse, cannot feel an itch on the other mouse. Okay, that just tells us it's not likely to be an itch, it's not likely to be peripheral nervous system, rather it has to do something with the brain. Okay? So if that's true, this gene should be expressed in the brain. And this was not expected because Hox genes are not expressed in the midbrain and forebrain, okay, to anybody's knowledge. So that was very disturbing. And here we actually show that in an adult mouse, If we look at the forebrain, midbrain, we can see mutant messenger RNA. What Joy did was to put a LOXP site in one. Hox genes are very simple. They just have 2 exons. So we could put a LOXP site in one to inactivate it, put a nonsense code on another one to make sure it's really not there. And then what that does is make the message a little bigger, so you can recognize the mutant RNA band. That's this one in the heterozygote, and here in the mutant homozygote, then you only have the mutant. Okay, so that tells us it is expressed in the brain of an adult mouse, where Hox genes shouldn't be expressed. So that was puzzling. It also tells us, because the signal here is about even, that it's not, cells aren't dying. It's not neurodegeneration induced by HOXB8, okay? The cells are still there. They're simply not doing something right that gives you then pathological behavior. Okay. So now, taking movies is very robust. But what Joy had to do is look frame by frame to see what that mouse is doing. So it's extremely labor intensive, a lot of work. So we switched to automation. What we have are platforms that are extremely sensitive to vibration. No matter what you're doing, you give off a different vibration pattern. If you're walking around or running or drinking or eating or grooming or scratching, whatever it is, you give out a different vibration pattern, and then this platform will pick it up. Then we'll have, essentially, algorithms that tell us what a particular vibration pattern is associated with what behavior. So that automatically then reads 24 hours a day, as long as you want, everything that mouse is doing, and also exactly where it is, and so on. Whatever that mouse is doing, we record it, okay? That makes us now, we can do everything automated, and we can start doing also pharmacology and so on. And it is robust. This is over 50 mice. And again, you can see mutants spend twice as much time grooming. And now the we is Shaquan, another graduate student in this case. Okay. Now I told you that, and I showed you that this gene, HOXB8, is expressed in the brain. However, the level of expression in the adult brain is low, and it's also salt and peppered. So it was difficult to identify essentially which cells in the brain were expressing HOXB8. And the way to get around that is simply to insert GFP into the sequence and then do a lineage analysis. So here we have essentially, again, the Rosa locus. We put GFP with a stop sign flanked by LOXP site, cross it to HOXB8 Cree, and then wherever HOXB8 is expressed, those cells will turn green and stay permanently green so we can follow them. Okay, so it allows us to see where in the brain cells are green. And this was an enormous surprise. The only green cells in the brain are microglia. So what I'm telling you is that microglia, the immune cells of the brain, are controlling behavior. Okay? And that's shown here, so those cells are green. CD11B is a pan-neuron marker for microglia, and here you can see coincidence of label, but not all microglia are green, okay? Turns out about 40% of microglia in the brain are green, that is express HOXB8, and 60% do not. There's 2 populations of microglia, one what we call the resident population, which actually is made during permitted hemopoiesis from the yolk sac, gets into the brain very early during embryogenesis, and a second population, which I'll show you now, gets into the brain much later, around birth. And here we're seeing essentially P2 means 2 days after birth. We start seeing green cells in the choroid plexus at the ventricular surface, and then 2 weeks after birth, now they're dispersed all over the brain. This is looking at the forebrain in this case. Okay, so we can see those cells and see what they're doing. Okay, if that's what I told you is correct, then it should be present in definitive hemopoiesis, so we can take blood, spread them out according to markers to distinguish myeloid B cells and T cells. Here's in the absence of Cree. In the presence of Cree, all of those populations move over. So that tells us that either this HOXB8 is expressed in hematopoietic stem cells or in progenitor cells capable of making both myeloid and lymphoid cells. Okay? Consistent with being in hematopoietic stem cells, we can now use different markers that specifically sort out stem cells, SCOV1 positive, c-KIT positive, in the absence and the presence of Cre. So that whole population then shifts over, telling us, indeed, they're likely to be hemopoietic stem cells. Now, this made an enormous, we simply show phenotype. That is, do the mutants show a phenotype with respect to microglia? Now, here we have a caveat. We used iris HOXB8 Cre, okay? And the reason we did that is we didn't want to destroy the HOXB8 function. But unfortunately, that also doesn't allow us to look at mutants, okay? Because one allele will always be wild type, okay, the one that has Cre. So we do see a reduction in number of microglia, but what we're doing is counting total microglia. If the other population compensated for reducing a HOXB8 population, then we may be underestimating how much this has been reduced. Okay, so what this tells us is it's reduced at least 15% and maybe more because we can't see mutants cells. Okay, we can't count them independently. But we have a very strong prediction, and that is we should be able to cure a mouse with a bone marrow transplant. Okay? That's our prediction. So, first of all, we just wanted to see whether we could actually get bone marrow into the brain. So we simply irradiated the mouse and then transplanted them with bone marrow that is labeled with GFP. And the answer is yes, we can get green cells in the brain. And here is 4 weeks after bone marrow transplant. You see buildup in the choroid plexus, ventricular surface. Then 12 weeks after bone marrow transplant. And this is about the same time course that you also see in human bone marrow transplants. It takes a long time. Then we get very good green cells in the brain. Okay? And we're getting about 75% transplantation, good transplantation. And here's the actual experiment. So here's a mutant mouse. We irradiate it to destroy its bone marrow, and then into that mutant mouse we put in normal bone marrow. Here we're looking about 3 weeks after the bone marrow transplant. If I showed you the same slide 2 weeks later, you would see a lot of lacerations and blood. However, in the presence of the wild-type bone marrow, all of those lacerations heal, all the hair grows back, and this mouse is permanently cured of that behavior. And here we're looking at how much time spent grooming, wild-type, mutant, and now after rescue with wild-type bone marrow. Okay? So that tells us we can cure that mouse with a bone marrow transplant. We can do the opposite. We can put mutant bone marrow into a normal mouse and give it the disease. Okay? So that it says it's both necessary and sufficient. Now there's a complication, and that is Hox genes turn on and off many, many genes. They're pleiotropic. They do lots of things. I mean, it's all the organs in that region, and you have a lot of different organs in that region, okay? And in particular, HOXB8 was also affecting sensitivity to pain, okay? And in fact, another group published that our results could be explained by that aspect of HOXB8 mutants. So we had to look into this. So let me go through that for you. So this is what it actually looks like. Here we're looking at a pan-neuronal marker and looking at a cross-section of the spinal cord. And if you actually counted nuclei and looked at them carefully, what you would see is there's less cells at the dorsal aspect of the spinal cord, and they're sort of not as organized as nicely as in the wild type. However, you can see that much more clearly if you use markers. And here we're looking at markers that are labeled the incoming sensory neurons, and then we're looking also at markers in the inner neurons that receive that input. Okay, so CGRP, calbindin, calretinin, and what you can see in the mutants, there are fewer cells and they're more scattered, rather than having nice laminar structures. Okay? But we can also do a behavioral assay, which is very simple and quantitative. You put a mouse on a hot plate and it jumps. So you can measure just how quickly does it react. And a wild-type mouse takes about a little less than 30 seconds, and a mutant mouse takes about twice to 3 times longer. That's the insensitivity to pain. And you can do the same kind of assays with noxious agents. Okay? And again, that's very quantitative. Okay, the first indication that we could actually separate those two phenotypes, the over-grooming removal of body hair, from this insensitivity to pain, was in the actual bone marrow transplant experiments themselves. So if we looked at those and now looked at the insensitivity to pain, what we can see, here's a wild type, here's a mutant, here's a rescued mutant, okay, but it doesn't affect insensitivity to pain. Whereas it was fully cured of the hair removal, okay? So that indicated to us that we should be able to remove those, separate those two by conditional mutagenesis, knock it out in the spinal cord and see what happens, knock it out in the bone marrow and see what happens, okay? For knocking it out in the bone marrow, we use Tie2 Cre. Tie2 is expressed in hematopoietic stem cells as well as in epithelial cells. Okay? So remember that as a caveat. But that's about the best marker that we could find that was pretty specific. Does it label microglia? And the answer is yes. So that says that we're labeling the hematopoietic cells and they're getting into the brain and so on. Okay? And here's the actual experiment. By knocking it out in the hematopoietic stem cells, we now still have removal of body hair. We get increased amount of time spent grooming. We do not affect insensitivity to pain. So we've been able, with Tie2 Cre, separate it in that direction. Okay? We affect the hematopoietic system, affect grooming behavior, but do not affect insensitivity to pain. If we look at the spinal cord itself, we can see essentially no effect with respect to the interneurons that are receiving those inputs. For example, here, compare that to here, compared to a mutant pattern, which is a lot less fewer and more scattered cells. So that tells us that we're not affecting the spinal cord. Okay. To do the opposite, now knock it out in the spinal cord and not in the hematopoietic system, we used a HOXC8, which is a parallel of HOXB8, and then the spinal cord has the identical expression pattern to HOXB8. Okay. And here this shows the expression pattern, and you could not distinguish HOXB8 from HOXC8. There are few green cells in the brain, but if we actually count how many green cells there are 15-fold fewer with HOXC8 compared to HOXB8. Okay? A few, but a lot less. Okay? And here's the actual experiment. There's no hair removal, doesn't affect time spent grooming, but now recapitulates insensitivity to pain. Okay, so then that tells us we've removed it, done it the opposite way, allowing us to separate essentially those two phenotypes and saying they're not overlapping with respect to cause. Okay. And again, if we look at the spinal cord, we get the mutant pattern rather than the wild type pattern in the spinal cord with HOXC8 as the Cre driver. Okay, so in summary, what I've showed you is that microglia are controlling behavior. Okay? So the next question is, how can microglia control behavior? How can they control neural circuits? And here there are many possibilities. Yeah, this is the actual circuit that we're talking about. This is called the OCD circuit in humans, and it's been worked out with functional MRI. Okay, involves the dorsal aspect of striatum, motor cortex, prefrontal cortex, thalamus, and it's the same circuit that is used to learn repetitive behavior. And then once you have repetitive behavior, it's also this circuit that then modulates that amount of repetitive behavior. Okay? Here we're showing microglia, and microglia sort of look like neurons. They have these enormous extensions, pseudopods, and they're waving around in space all the time. Okay? So one possibility is, for example, we know that microglia produce cytokines. We know they produce chemokines. We know they produce neurotransmitters, for example, glutamate. So they could be producing chemicals that could in turn then affect neurons, which then affect that particular circuit. So that's one possibility. The other possibility is actually more interesting, and that is these pseudopods. Okay, what you can see is, first of all, they're weaving around in space, and what you can calculate that in 1 hour, they can actually sense the entire parenchymal surface of the brain, because there's a lot of microglia. Okay? The other thing that Waki et al. Has shown in Japan is that these things are waving around, then all of a sudden they stop. And then they stay there 5-10 minutes. And the question is, what are they doing? And if you look, what they do is they actually wrap that pseudopod around a synapse itself. Okay? Intimate contact. By electron microscopy, those membranes are right on top of each other. Okay? So the question is, why would you go to all that trouble unless you wanted to control activity across that synapse? So another possibility is that they're actually directly talking to each other and can modulate electrical activity across those boundaries. Okay? Now, the technology is there right now to be able to prove that, and that is optogenetics. Okay? You have channels that you can turn on and turn off with light. And you have channels, for example, that pump sodium, activate cells. You can have other channels that actually activate chloride and silence cells. And the response times is in nanoseconds, okay? That means you can get information. You can pulse as particular frequencies and then ask if we activate microglia, do neurons all of a sudden pick up that activation, and it's the same pattern that we're feeding it. We can reverse it. We can activate the neurons and ask neighboring microglia, do they now receive signals? So we can start asking questions. Do they talk to each other? How do they talk to each other? And how do microglia control behavior? And all of those experiments are ongoing. Unfortunately, I don't have the answer, but I think it's an interesting story. But I'll point out a couple of other things. First of all, if you look at any neuropsychiatric disorder, a monopolar depression, bipolar depression, schizophrenia, autism, OCD, OC spectrum disorder, even schizophrenia. Okay, all of them have been long known to be associated with immune dysfunction. 30 years of experiments. What wasn't known is cause and effect. Okay, am I depressed and therefore my immune system isn't working well, or is my immune system defective and therefore the risk of depression goes up? Okay, which is cause and which effect? In this particular system, we have the first system where we know cause and effect. Defective microglia are giving rise to a pathology, behavioral pathology. It's very common. 3% of people have it. So in any population, it's been looked at all over the world, all populations, 3% have OCD, 3% population have trichotillomania. Very common. So what we're arguing is we know a lot more about immunology than we know about how our brain works. If you treated the immune dysfunction, could you now have an effect, essentially, on the behavior? Okay? So that's one problem that one should be thinking about. The other is bone marrow transplants. 600,000 bone marrow transplants have been done approximately worldwide. All of these diseases are 1%, 2%, 3% of the population. Okay, enormous overlap. So you could ask, well, people have done the experiment, what happened? So I've called all of these centers, not all, but a lot. And they say, unfortunately, we do not keep records. However, I have a stack of emails about that high from patients saying very remarkable things have happened after a bone marrow transplant, okay? Which is interesting. So I think that's one thing to also think about. Another thing is, what I've been talking about is all in mice. It's a correlation of behavior. It looks like OCD, it looks like trichotillomania, but what is the real correlation? Now, one thing that's you have to think about is we're looking at HOXB8 mutant homozygotes. A recessive mutation, we have to have 2 defective genes. Okay, 3% of the population are not going to be homozygous in all populations for any gene, which is fortunate. Okay, don't inbreed that much. However, if you have something that's common, there's a couple of ways of explaining it. One is that there's a lot of genes that can give rise to the same output. The other possibility is there is essentially a polymorphism that's very common in the human population. Turns out in HOXB8 there is. Very common. 20% of the people have this polymorphism. If you then look to see whether they have trichotillomania, it tracks with the mutation. Okay? So that says it's, what we're looking at in mice is maybe also applicable to humans. That polymorphism isn't in the coding region. It's in essentially in an intron, but if I look at it, I would say it's likely to be a splicing mutation. We can prove that by simply taking that polymorphism, sticking it into mice, the human polymorphism, sticking it in mice, and asking what happens. Is splicing interfered or not? Those experiments are ongoing. So I think we have now a way of essentially starting to look at the human population and ask, do they have defects in HOXB8? And what you have to argue is you're not going to see 3% of the population is homozygous for this, even this polymorphism. However, what you can say is that you could have the polymorphism if it does reduce the function of HOXB8, couple it with another mutation, which then together would be equivalent of a HOXB8 homozygote. What are those other mutations? They're going to be something to do with the immune system. Lots of genes affect the immune system. Okay, so coupling them together, then all of a sudden could give you that spectrum. And so obviously now we can start looking at humans. 3%, it's easy to get DNA. So at that point, I'll stop and open it up for questions, and thank you. Yes? Okay, back there, and then, okay, yes? Uh-huh. Uh-huh.
My question is that which part of the brain or cortex this microglia are more found?
Uh-huh. Good.
Especially the sensory cortex, motor cortex, and the distribution of the microglia.
Yeah. That's this slide. Go ahead. Good. Very good question. I didn't plant this. Okay. So this actually looks, first of all of this red, these are fibers coming from the spinal cord, and they express high levels of HOXB8. But what we're interested in is you would be able to see red and green ones, okay? The red ones are HOXB8 expressing microglia. The green ones are all other microglia, okay? And so if you look at that ratio, okay, that circuit that I just talked about right here, okay, motor cortex, dorsal aspect of the spinal cord, and the frontal cortex, that expresses high levels of HOXB8 microglia. Whereas in other regions, for example, here and here, where there isn't that circuit, then there are low levels of HOXB8 expressing microglia. So that says that the HOXB8 expressing microglia are in the right place to do what they're supposed to be doing, controlling that circuit. And so this may be a coincidence, but it is an interesting coincidence, and also tells us that these cells, the microglia, know where to be in space. It's not that they're randomly in space, but they actually have a distribution pattern, which is interesting with respect to pathology. Okay, so that's the answer to your question. Okay. Yes.
We have time.
What are the differences in behavior that you've seen after that patients have reported after bone marrow transplants?
You can go both. You can go either way. The other thing, and so you can get patients that said, I used to have it and now it's gone, and you have patients that didn't have it and now they have a pathology.
Are you talking about obsessive compulsive disorder? Okay.
Yeah.
Or just staying in the shower a long time?
No, this is OCD. Yeah. And autism is also included in that because a lot of aspects of autism are actually OCD-like. I mean, so there's a lot of comorbidity with respect to the output of behavior. What I should say, the other thing I should say is that, I don't personally believe that this system, this microglial system, was designed to control trichotillomania. If I were up there, I just wouldn't do that. Okay, it seems too complicated. What are we really looking at? And I think what we're really looking at, if you look at any neuropsychiatric disorder, the other thing that's often associated with is anxiety. If you ask any OCD patient, I've asked thousands, if you are in what happens when you're anxious? And they say, when I'm anxious, I go into my pathology. When I'm relaxed, I don't do it, okay? Anxiety. Yeah, so I think what this system is really being made to do is control levels of anxiety, okay? And that once you get out of whack and now you're anxious all the time, then you have a pathology. And the output can be different depending on other genetic factors, okay? And also environmental factors. So the output could be trichotillomania, or the output could be monopolar depression or whatever, okay? But it's related essentially to high levels of uncontrolled anxiety, and that's what the system is made. Remember that, anxiety is, it's a very primitive behavior. Actually, grooming is a very primitive behavior. I already indicated that all animals groom. And when you're, if a tiger is chasing you, you want to run, okay? That's, boom, that's a good response. On the other hand, if two people want to communicate, what they have to do is reduce their level of anxiety, have some trust, and then be able to communicate with each other. Okay, so that's where you want to then be able to modulate the level of anxiety depending on the input. Yes?
I have a question about microglia, which are found throughout the body.
Mm-hmm.
Found in what behavioral people are beginning to talk about as fascia.
Mm-hmm.
And the question is whether or not any of these patients report in that they also have. Sorry, the question would be whether or not any of these patients who report in with changes, whether they also have, I'm going to call it generalized arthritis, or things of that sort, which would turn out to be immobilization mechanisms that they never had before, or whether they would lose this kind of thing with this bone marrow transplant.
Right. I mean, I'll give you a sort of a longer answer than you want. Microglia are doing a bunch of things, okay? One of the things they're important for is if you do have an infection, an infection in your brain or you have a stroke, they go there and clean up the mess. So that's one function of microglia. A second function of microglia is actually pruning. Every connection we make, every synapse we make about twice as many as we need, and then there's a time period where they go and remove half of those, and then usually, hopefully, remove the bad half, the ones that are strong, and therefore, and then leave the others. Okay, so that's another mechanism of this type that could affect behavior. Is this simply that you're not now doing the pruning that you should be doing, and so on. And then finally, what I'm saying is there's also possibility of direct electrical interaction. I mean, the microglia themselves are really only in the CNS. But the monocytes, which it's the precursors of microglia, were in the periphery, then they're everywhere. And nobody actually so far, I mean, that one thing we would love to have is a marker that distinguishes microglia from monocytes. So far, nobody's ever reported such a marker. Monocytes are macrophage precursors, essentially. And so they would be affecting arthritis and all those kind of things, and big players in that. But the CNS equivalent of that is microglia. But then once they get into the brain, once they get into the CNS, they turn on a new program, and they look quite different from monocytes.
I'm going to thank Dr. Capecchi for a most